This recording is part 3 of 3 for Chapter 2, the chemical level of organization. We know that organic compounds are going to contain at least one atom of carbon and one atom of hydrogen. Large organic compounds are referred to as macro, which means huge molecules. These large organic compounds, or macromolecules, are unique to living systems. They often have long chains of carbon backbones where carbon is covalently bonded to carbon, is covalently bonded to another carbon, etc. And carbon forms covalent bonds. It doesn't lose or gain electrons to form ionic bonds, because carbon has 4 electrons in its outermost valence shell. It's tough to lose 4 electrons or gain 4 electrons. It's more likely to share electrons with others. That's why carbon tends to form covalent bonds. There are 4 main types of organic compounds, including carbohydrates, lipids, proteins, and nucleic acids. Some texts will include adenosine triphosphate, that's what you see at the bottom of the slide, as ATP, as a 5th type of organic compound. It is organic, but it varies text by text. Know at least carbohydrates, lipids, proteins, and nucleic acids. Because macromolecules are made up largely of carbon backbones, in order for those molecules to interact with other molecules or compounds, we often find functional groups on the ends of these macromolecules. We've already seen one of these functional groups at work. That was the hydroxyl group that played a part in dehydration synthesis and hydrolysis. You'll notice to the right of the hydroxyl group at the bottom is a carboxyl group. Carboxyl groups, as you could imagine, can also easily play a role in dehydration synthesis and hydrolysis, as well as the amino group. Not pictured in this particular view is a methyl group. Methyl groups can be found on the ends of amino acids. They can be found on the ends of fatty acids in lipids. And to the right of the amino group in this picture, you'll see a phosphate group. Phosphate groups are found in phospholipids and nucleotides. They, as well as hydroxyl groups, are also polar, and phosphate groups make up a huge part of adenosine triphosphate, which has 3 phosphate groups. When the 3rd phosphate group from adenosine triphosphate is broken off, when that bond is broken, energy is released to do work. That's why adenosine triphosphate is a very big contributor to cellular energy. Carbohydrates are an important source of energy. Carbohydrates include compounds such as sugars and starches. They contain carbon, hydrogen, and oxygen at a 1:2:1 ratio. Carbohydrates are actually a big part of macromolecules such as adenosine triphosphate, ribonucleic acid, and deoxyribonucleic acid, as well as glycoproteins, the term which literally means sugar protein, glycolipids, which are sugar lipids, and other molecules within the body. Polysaccharides, poly meaning many, sugars, saccharides, are important in the storage of energy, as is glycogen. Glycogen is a molecule that is stored and found in the liver as well as in muscle cells. Polar compounds, such as different types of sugars, will dissolve in water and are easily transported through the blood. You'll notice at the bottom a picture of cellulose. Cellulose is a type of carbohydrate, and our bodies do not have enzymes to break down cellulose. Let's take a look at the chemical composition and structure of carbohydrates. To the left we have monosaccharides, mono meaning one, saccharide meaning sugar, and these single sugars include glucose, fructose, galactose, deoxyribose, and ribose. You'll notice that these monosaccharides are split into two groups, the hexoses and the pentoses. Hexose monosaccharides contain 6 carbons. Pentose monosaccharides contain 5 carbons. On the right side, you'll notice the disaccharides. Disaccharide sugars are formed when 2 monosaccharides are joined by dehydration synthesis. Here we have a disaccharide known as sucrose. Sucrose should sound familiar to you because it's table sugar. Sucrose is formed by joining together glucose and fructose by way of dehydration synthesis to form that disaccharide. Lactose is a disaccharide formed by the joining of galactose and glucose. When we form maltose, that is the product of joining 2 glucose monosaccharides together. Again, monosaccharides are simply simple sugars. They include glucose, fructose, and galactose, and deoxyribose as well as ribose. In order to make bonds to go from monosaccharides to disaccharides, we have to have an input of energy. Disaccharides are 2 sugars. These, again, are composed of 2 monosaccharides being joined together by way of dehydration synthesis. They can be broken down by way of hydrolysis, and disaccharides are too large to pass through membranes of cells, which is why they would have to be broken down into their monosaccharide components in order to get into cells. From monosaccharides and disaccharides, we go into polysaccharides, which means many sugars. Polysaccharides are larger, much more complex carbohydrates that have been linked together by way of dehydration synthesis. There are 3 kinds of polysaccharides, including cellulose, starches, and glycogen. Cellulose is a carbohydrate form found in plants that our bodies cannot digest because we don't have the enzymes to break down cellulose. That's why it's often referred to as fiber. And when we eat foods that have a lot of cellulose in them, the fiber helps to sweep through our digestive system and clean it out. Starches are carbohydrates that are formed by plants that we can break down, and these are found in things like grains and potatoes. Glycogen is how our bodies store excess carbohydrates, and glycogen is specifically stored in our bodies in skeletal muscle cells and liver cells. Glycogen can be broken down by way of hydrolysis in order to use energy when it's needed. Carbohydrates are not only important because they serve as a source of energy for the cells, they're also important because they have a structural purpose inside and outside the cell. You'll notice DNA to the left, which contains a sugar phosphate backbone on either side of the double helix. We'll look at that a little bit more closely later. And if you look closely at the picture to the right, you'll notice that there are glycolipids in the cell membrane as well as a glycocalyx on the surface of the cell membrane. Let's see if I can, here's that word, glycocalyx, which means sugar coat. Here's a glycoprotein, which is a protein that has a carbohydrate bonded to it. So those are some examples of how carbohydrates can function structurally. By the way, the glycocalyx, which is the sugar coating candy sweetness on the outside of the cell membrane, is what helps your body determine self from non-self. Cells will often look at the glycocalyx markers on the surface to tell whether or not that cell belongs to your body. Lipids are macromolecules that are insoluble in water but will dissolve in alcohol and ether. Lipids contain carbon, hydrogen, and oxygen. Their carbon to hydrogen ratio is 1:2, and these are important molecules because they help to form structural elements of all the cells. They also act as energy reserves. They help provide insulation, protection, and form hormones in our body. In the blood, we know that lipids can combine with proteins to provide lipoproteins for transporting lipids through a watery medium. Triglycerides are neutral fats which are made up by joining 3 fatty acids to a molecule of glycerol, again by dehydration synthesis. We can see the glycerol molecule here to the left, and you'll notice that each carbon atom has 4 covalent bonds, because carbon likes to form 4 covalent bonds. And on the right side of the glycerol molecule, each oxygen molecule to which is bound by carbon on the opposite side has an atom of hydrogen. If this hydrogen is removed and this oxygen and hydrogen is removed from this carboxyl group on a fatty acid chain, that's where we get our dehydration synthesis. So in order to attach these glycerol molecules to 3 fatty acids, that's where we get triglycerides, 3 fatty acids attached to the glycerol. We take away the hydrogen and the oxygen for each of these 3 sites, and that is going to form a triglyceride and 3 molecules of water. It's put together by way of dehydration synthesis. Remember, this is reversible, so the triglyceride can be broken down into a glycerol molecule and 3 fatty acids simply by hydrolysis, adding water to break or burst apart. Now, that would require some enzymes, since we know that our triglycerides do not dissolve in water. And fats are important because not only do they insulate our bodies and provide cushioning, but they also help store energy. Fat molecules can be cycled into producing energy for our bodies. Triglycerides are nonpolar, and that means that they are fat-soluble, often transported throughout the body as lipoproteins since they don't dissolve in water. Triglycerides are made with a glycerol and 3 fatty acid molecules. When we look more closely at fatty acids, we can see that there are some fatty acids that are saturated and some that are unsaturated. The fatty acids that are saturated have as many hydrogen atoms as can possibly be bonded to each of the carbons in that fatty acid chain. So in the top picture, we see, disregarding the carboxyl functional group, we should see that each carbon in the chain has as many possible bonds to hydrogen as it can have, all the way to the end, where we have this last carbon bounded to 3 hydrogens. Remember, carbon wants to form 4 covalent bonds because it's got 4 electrons in its outermost valence shell. It would like to have 8, so we need 4 more bonds covalently to make that happen. That is a saturated fatty acid. Well, what's the difference between a saturated fatty acid and an unsaturated fatty acid? Unsaturated fatty acids occur when there is a double bond between 2 carbon atoms. Remember that carbon wants to have 4 covalent bonds. A double bond is another way of creating a bond so that there are still 4. If we look at this carbon and count the bonds, we still have 1, 2, and we count the double bond as 3 and 4. This carbon over here has 2 in the double bond that it shares, 1, 2, and 3, and 4. So these carbon atoms are fulfilling that octet rule of wanting to have 8 electrons shared in their valence shell. However, the double bond means that there are not as many possible hydrogen atoms in an unsaturated fatty acid as there are in a saturated fatty acid. Saturated fatty acids are saturated with hydrogen. Normally, when people eat unsaturated fats, that's considered a much healthier diet. Other types of lipids include phospholipids, where we have a phosphorus-containing group, which, by the way, is polar. Anything that is polar is going to be water-loving or hydrophilic. Hydrophilic means water-loving. The phosphorus-containing group of the polar head is joined to a glycerol backbone, which in turn has 2 fatty acid hydrocarbon chains attached to it. By the way, the term hydrocarbon means a chain of carbons and hydrogen. So the fatty acid chains are water-fearing because they are the lipid component of the phospholipid molecule, and fat and water don't mix. So the nonpolar tail is hydrophobic, water-fearing. Phospholipids are a major component of the cell membrane. This takes us to steroids. Sterile molecules consist of 4 hydrocarbon rings. Again, the word hydrocarbon just means hydrogen and carbon. And these sterile molecules have both polar and nonpolar regions. Examples of steroids include cholesterol. Certain hormones are sterile hormones, and we will see these steroid molecules embedded in the cell membrane to help stabilize it, and they're also found in bile that is produced by the gallbladder. We also have lipid molecules known as prostaglandins. Prostaglandins are lipid molecules that are chemical messengers. They'll actually signal certain things in our bodies to help regulate things like our blood pressure, our temperature. They can also regulate and signal pain sensation and inflammation. Sometimes prostaglandins can act as protective molecules. For instance, we have a special prostaglandin in our stomach known as prostaglandin E that helps protect the lining of our stomach against the hydrochloric acid. Trans fats. These are common in synthetic oils, and because they're in synthetic oils, they'll be found in processed foods like baked goods, fried foods, snacks. These are fatty acids that have oils that have been modified, where the natural fatty acid or hydrocarbon chain would contain a double bonded carbon in order to produce a trans fat or partially hydrogenated oil. That's another name for these. What manufacturers will do is they will apply heat to break the double bond and force hydrogen ions on that carbon where it did not normally exist originally. And trans fats are not good for the body. They tend to raise the amount of low density lipoproteins, which are LDL. That's considered harmful cholesterol, and they will lower the amount of high density lipoprotein, which is the good cholesterol in your body. One of the biggest problems with saturated fats when they've been made artificially is when they are ingested, the hydrogen atoms, which are ions, when they break apart off of this saturated fatty acid, act as free radicals and can damage the endothelium of blood vessels and the heart. This takes us to proteins. Proteins contain carbon, hydrogen, oxygen, and nitrogen. Proteins have a bunch of functions in the human body. They can act structurally, they can help different things move and contract and expand. They can help transport things, assist in moving things that are not normally soluble, like lipoproteins. They can act as buffers to prevent changes in pH. Proteins are also able to help as chemical messengers. There are proteins that act as hormones, as well as lipids that act as hormones. Proteins can act as antibodies to help protect us against diseases. And all enzymes are proteins. Not all proteins are enzymes, but all enzymes are proteins. Proteins are made up by amino acids. There are 20 different types of amino acids that form building blocks for proteins. You're going to need to know that for microbiology, should you go into microbiology, that there are 20 different amino acids. Each amino acid, in terms of structure, has a central carbon bonded to an amino group on one side and a carboxyl group on the other side. That gives us two functional groups. What changes the amino acid, because we said there's 20 different types, is dependent upon the side chain that's bonded to the central carbon. The side chains vary in terms of their chemical structure and therefore will give this amino acid different properties depending on what is bound there. Amino acids are joined together with one another to form a chain of amino acids by way of special bonds that are known as peptide bonds. Amino acids are the building blocks of proteins. So as we build a building block of amino acids, we're building peptide bonds to form a primary structure of a protein. And the recipe for all the proteins in our bodies is stored on our DNA. That's what DNA is. It's the instructions for how to build proteins. Peptide bonds are formed by dehydration synthesis. They can be broken down by hydrolysis. Amino acids can be joined together by dehydration synthesis. Here we have an example of glycine being joined to alanine by removing an oxygen and hydrogen off of the carboxyl group from glycine and a hydrogen off of the amino group from alanine. When dehydration synthesis occurs and these atoms are removed, carbon will form a bond directly with nitrogen. I'm going to draw it up here. It's a single bond. I don't like the picture down below because that pole looks like a double bond, and it's not. It's a single bond. So this is a single bond between carbon and nitrogen that forms after dehydration synthesis. And again, the product is a dipeptide. 2 amino acids joined by a peptide group forms a dipeptide, and we also have water as a result in this process. If we had 3 amino acids joined together, it would form a tripeptide. Many amino acids makes a polypeptide, and more than 50 amino acids is the beginning of our protein. Proteins, as they're being formed, go through a process of turning into different structures. The initial chain of amino acids is referred to as the primary structure of a protein. It's just simply a chain of amino acids. Because of the different types of atoms and hydrogen bonds, remember hydrogen bonds are important in developing structures, those hydrogen bonds will cause the primary chain of amino acids to start developing into a secondary structure of protein by either turning into what's known as an alpha helix coil or a beta pleated sheet. If you've ever taken a piece of paper and folded it back and forth to make a fan, that's what a beta pleated sheet is like. And you'll notice the tiny little dots between these amino acids are hydrogen atoms being attracted to other atoms that have a partial charge within the amino acid. And that's what helps with its structure. From the secondary structure of a protein, which we said was either an alpha helix coil or a beta pleated sheet, the protein, as it develops, continues to fold in on itself, following a globular unit called a tertiary meaning third level, tertiary structure. And then when 2 or more tertiary structures form together, we have a quaternary structure. Hemoglobin is a great example of a quaternary globular protein where 4 tertiary structures join together, each with a heme group containing iron to help transport oxygen. Now, some proteins are globular proteins where they look like a glob. Other proteins develop more as fibrous proteins, so they look more like fibers. Here's an important question. What happens if we mix up the order of the amino acids in the primary structure? If the order of amino acids gets mixed up, that's going to affect the structure of the protein, and that can cause a problem in the function, because remember, structure and function are interdependent. So if the structure is mixed up or messed up, then potentially that could cause misfunction in the human body. Remember, all of this information is coming off of the DNA. What happens if there are problems in the DNA? Proteins can undergo a process of unfolding. All of that we just saw in the previous slide can unfold and unravel and come apart through a process called denaturation. You have already seen proteins denature if you've ever fried an egg. Normally, the albumin, which is the clear part that turns white, normally that's clear protein. However, when exposed to extreme changes in temperature, proteins can denature. Now, it's not just severe changes in temperature. Severe changes in pH can also cause proteins to denature, as well as severe electrical input can cause proteins to denature. Now, sometimes denaturation is reversible, sometimes it is not. So it depends on how far the protein is denatured through those different levels of structure. Remember, we went from primary to secondary to tertiary to quaternary. So depending on how much is denatured, this may not be reversible. We said that some proteins act as enzymes. When proteins act as enzymes, they usually need some assistance, which comes in the form of a cofactor, which is often a mineral like copper or iron, or they may require a coenzyme, which is often a part of a vitamin. Enzymes act on molecules called substrates. When substrates bind to enzymes, there is a specific requirement because of the shape of the substrate having to fit into the receiving end of the enzyme, just like a lock and a key. When the substrates bind to the enzyme assists in forming a product by helping the chemical reaction to occur. And in forming this product, the product is then detached from the enzyme, so the enzyme can repeat the process. We said before that the enzyme acts as a catalyst to speed up the rate of chemical reactions. When the substrates match the enzyme shape, the specific parts of the substrate that need to interact with one another are approximated in such a way that it allows that chemical reaction to occur much more easily and quickly. You'll notice that after the product detaches, the enzyme is still available to act on new substrates. So the enzyme never gets used up as a catalyst. It continues to work again and again as substrates are available. We have said previously that enzymes do what? Do they lower the activation energy or do they raise it? Well, they actually lower it because they decrease the energy of activation to take us from substrate to product. Are enzymes changed by the reaction they catalyzed? Nope. Every chemical reaction is going to require some kind of energy for the reaction to occur. We call this activation energy. And this is the energy that's required to either break the bonds of the reactants so they can rearrange themselves and become the product, or put things together, because putting things together requires energy. So ultimately, the randomness of reaction is decreased temporarily by presenting the proper parts of the substrate to one another so that the reaction can occur, making bond breaking or bond forming easier. Enzymes are unchanged. They can go again and again and again, so only small amounts of enzymes are needed. Most enzymes in our bodies can catalyze millions of reactions per minute. Nucleic acids are the largest molecules in the body. They're made up of carbon, hydrogen, oxygen, nitrogen, and phosphorus. The structural units of nucleic acids are referred to as nucleotides, each nucleotide containing a nitrogen-containing base, a pentose sugar, and a phosphate group. There are 5 nitrogen-containing bases in nucleic acids: adenine, guanine, cytosine, thymine, and uracil. Nucleotides are made up of a pentose sugar, a phosphate group, and nucleic acids. They're broken down into 2 groups, pyrimidines and purines. I don't care that you memorize which belongs in which group. That's neither here nor there. One thing to know is that the sugar in DNA is deoxyribose, whereas the sugar in RNA is ribose. Now, it says on this picture DNA. That's incorrect. It's actually RNA. So what's the difference between deoxyribose and ribose? The deoxyribose sugar actually has 1 less oxygen than ribose. So here we have our nucleotides linked together with a phosphodiester link, and here we have our pentose sugar, our phosphate group, and our nitrogenous base. Now, the nucleic acids, you're going to notice that in DNA we have adenine, thymine, cytosine, and guanine. In RNA we have adenine, uracil, cytosine, and guanine. So thymine is only in DNA, uracil is only in RNA. And when things match up, we're going to see how these match up soon, cytosine and guanine will always form a bond with one another, whether cytosine comes first and matches with guanine, or guanine comes first and matches with cytosine. In DNA, adenine and thymine will always go together, and in RNA, adenine and uracil will always go together. We'll see that a little bit more closely. DNA is important because it's got all the information on how to build a protein. 3 nitrogenous bases in a row on DNA are going to have the code for 1 amino acid to be part of that building block of a protein. And there are 46 strands of DNA in every human cell that hold the primary structural codes for all of your proteins, including proteins that act as hormones. Hemoglobin is a protein. Remember, we said all enzymes are proteins. Collagen is the most abundant protein in the human body. That's important for you to know. Collagen is the most abundant protein in the human body. Muscle fibers contain proteins. There are regulatory proteins that allow things in or out of the cell. So DNA is the instruction manual for each protein in your body, and DNA makes you who you are. We talked about the fact that there are 20 different amino acids involved in producing proteins. I actually have them listed on this slide. The essential amino acids are ones that our bodies can't make, and they have to be consumed in our food. The non-essential amino acids are things that we can produce if we have enough nutrition and not too much stress. At the top of this picture, we see deoxyribonucleic acid, otherwise known as DNA. You'll notice that the structure is that of a double-stranded helix, and you'll also notice that it contains 4 nitrogenous bases that are complementary to one another. Adenine and thymine are complementary nitrogenous base pairs that form links together by way of 2 hydrogen bonds, whereas cytosine and guanine will form bonds together because they have 3 hydrogen bonds that can form between them. Ribonucleic acid, which is also known as RNA, is a ribose-containing nucleotide that can help us produce proteins from the information stored on DNA when it acts as messenger RNA. RNA contains cytosine and guanine just like DNA. However, RNA does not have thymine, it has uracil, and that's going to be important when we look at how the information is taken from DNA and written onto a strand of messenger RNA. Here we have a slide showing some of the differences between RNA and DNA. Again, RNA in this picture is shown as a single strand. RNA comes in different forms. We'll see those a little bit later. RNA contains ribose, where DNA contains deoxyribose. RNA contains uracil, DNA contains thymine. RNA is much smaller, anywhere between 100 to 50,000 nucleotides, where DNA may have 45 million nucleotides. RNA helps protein synthesis that's directed by the DNA, and DNA is important because it stores that genetic information that controls the synthesis of proteins. Adenosine triphosphate is important because it is the source of cellular energy, and that helps us transport things throughout our cells, throughout our bodies. It helps our muscles contract, and it's important for a lot of anabolic reactions. Remember, anabolic reactions require energy, whereas catabolic reactions release energy. If we look closely at adenosine triphosphate, you'll notice that it is made up of a molecule of adenine. It also consists of a ribose sugar and 3 phosphate groups that are held together with high energy bonds. The last bond between the outermost phosphate groups has the highest energy, which can be hydrolyzed. Remember hydrolysis, to use water to break or burst apart. And when this bond is broken, energy is released to do work. Here's a little review for you to do at your leisure. And that concludes this recording of part 3 of 3 for chapter 2.